In the grand narrative of scientific breakthroughs, the discovery of DNA's double helix stands as a monumental peak, a moment that fundamentally changed our understanding of biology, genetics, and life itself. The names most famously associated with this peak are James Watson and Francis Crick. But the terrain of this discovery was mapped. The crucial path to the summit was cleared by an extraordinary scientist whose contributions were, for decades, tragically understated. Her name was Rosalind Franklin. To understand the story of DNA, you must first understand her. Born in 1920 into an affluent and intellectual British Jewish family, Franklin was fiercely intelligent and determined from a young age. She excelled in the sciences, a field not always welcoming to women, and earned her doctorate in physical chemistry from Cambridge University in 1945. Her early work wasn't on DNA, but on the microstructure of coal and carbon, research that proved vital to the British war effort and established her as a world-class expert in a challenging technique, X-ray crystallography. Imagine trying to determine the intricate three-dimensional structure of an object so small you could never see it, even with the most powerful light microscope. This is the challenge of crystallography. The technique involves shooting a beam of X-rays at a crystallized substance. As the X-rays pass through the crystal, they are diffracted or scattered by the atoms within it, creating a complex pattern of spots and smudges on a photographic film. A skilled crystallographer, through painstaking mathematical analysis, can then interpret this pattern to deduce the precise arrangement of atoms in the molecule. It is a field that demands immense patience, mathematical prowess, and technical perfection. After honing these skills to a razor's edge at a prestigious state laboratory in Paris, Franklin was offered a research fellowship at King's College London in 1951. The mission? To use her expertise to study the structure of DNA. This was the frontier of biology. Scientists knew DNA carried genetic information, but how it did so was a complete mystery, a mystery locked within its physical shape. Upon arriving at King's, however, Franklin stepped into a situation fraught with ambiguity and tension. The project was overseen by John Randall, but the DNA work was to be shared with another scientist, Maurice Wilkins. A misunderstanding from the very beginning would poison their professional relationship. Wilkins, who was away when Franklin was hired, returned to view her not as an independent peer, but as a technical assistant assigned to his project. Franklin, rightly, saw herself as the sole authority over her own research domain. This clash of personalities and expectations created a fractious environment, one where collaboration withered and communication broke down. Undeterred by the chilly atmosphere, Franklin set to work with her characteristic rigor and precision. She refined the equipment, developed new techniques for preparing and hydrating the delicate DNA fibers, and began to produce X-ray diffraction images of unprecedented clarity and quality. It was a testament to her exceptional skill. Her meticulous approach soon led to a monumental discovery that had eluded Wilkins and others. She determined that DNA could exist in two distinct forms, depending on the level of hydration in the sample. She named them the A form, which was drier and more crystalline, and the B form, which was wetter and what we now recognize as the primary form in living cells. This distinction was absolutely crucial. Previous, blurrier images had been a confusing mix of both forms. By separating them, Franklin could get a much clearer picture of each. And it was her work on the B form that would lead to the breakthrough. Over a period of more than 100 hours of X-ray exposure in May 1952, a single image was captured, a pattern of dark spots on a piece of film that would become one of the most important photographs in the history of science. It is known simply as Photo 51. To the untrained eye, it looks like a fuzzy black and white X, but to Franklin, it was a revelation, a Rosetta Stone for the molecule of life. The distinct X shape was the unmistakable signature of a helix. The regular spacing of the smudges or layer lines along the arms of the X allowed her to calculate with precision the dimensions of the helix, its angle and its pitch. Even more subtly, she noted that the fourth layer line was missing. A student of crystallography would know this was a telltale sign that the helix was not single, but a double helix, with its two strands intertwined and slightly offset. Furthermore, the intensity of the spots at the very top and bottom of the image told her that the phosphate sugar backbones, the structural supports of the molecule, had to be on the outside, with the nucleotide bases A, T, C, and G tucked away on the inside. In her lab notebooks, Franklin recorded these conclusions. She had the fundamental data, the hard evidence that defined the double helix. The question that has haunted history ever since is, with this treasure trove of information in her possession, how did she become a footnote in her own discovery? The answer is a complex and controversial tale of ambition, back-channel communications, and a scientific race that was about to reach its finish line, leaving its most vital contributor behind. While Rosalind Franklin was painstakingly collecting her data at King's College, a different kind of effort was underway 50 miles north at Cambridge University's Cavendish Laboratory. There, a young, brash American biologist named James Watson and a brilliant, slightly older British physicist named Francis Crick were also obsessed with DNA. But their approach was different. They weren't conducting their own experiments to generate new data. Instead, they were building physical models, like a complex Tinker Toy set, trying to fit together what was already known about DNA's chemical components into a structure that made sense. Their initial attempts had been failures, in part because their information was incomplete and, in some cases, incorrect. They knew the prize was immense. Whoever solved DNA structure would be immortalized. And they were in a race, not just with the team at King's College, but also with the world-famous chemist Linus Pauling in California. They were desperate for a breakthrough, a key piece of evidence that would put them on the right track. That key piece of evidence was sitting in Rosalind Franklin's desk drawer. The controversy ignites around how they got it. In late January 1953, James Watson visited King's College. There, he met with Maurice Wilkins. In a moment that has become infamous in the annals of science, Wilkins, without Franklin's knowledge or consent, showed Watson photo 51. For Watson, who had seen Franklin present some of her data before but had not grasped its full significance, this was a lightning bolt. He later wrote in his book, The Double Helix, that the instant I saw the picture, my mouth fell open and my pulse began to race. The clear X pattern screamed helix to him in a way it hadn't before. He now had a visual confirmation of the molecule's fundamental shape. But that wasn't the only piece of Franklin's work that found its way to Cambridge. In a separate, equally troubling breach of scientific etiquette, another scientist at Cambridge, Max Perutz, who sat on the Medical Research Council committee that reviewed Franklin's work, gave Watson and Crick a copy of an informal report Franklin had written summarizing her findings for the committee. This report was not a published paper, it was confidential. And it contained her precise, hard-won calculations and crystallographic data, including her conclusion that the DNA molecule belonged to a specific crystal symmetry group that strongly implied it was composed of two chains running in opposite directions. Armed with these two critical pieces of Franklin's data, the visual proof from photo 51 and the precise mathematical parameters from her report, Watson and Crick had everything they needed. The data acted as the rigid constraints for their model building. They knew it had to be a helix, they knew its precise dimensions, they knew it had to have the phosphate backbones on the outside, and they knew it was a double helix with anti-parallel chains. Within weeks, they had solved the puzzle, building their iconic, elegant double helix model, where adenine, A, always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. In April 1953, their groundbreaking paper was published in the journal Nature. In the same issue, separate papers by Wilkins's team and by Franklin and her student Raymond Gosling were also published. But the positioning was key. The Watson and Crick paper came first, presenting the model as the definitive discovery. The other papers were presented as supporting evidence, as if their work was done to confirm a pre-existing theory. Their paper contained a now famous understated line. We have also been stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M.H.F. Wilkins, Dr. R.E. Franklin, and their co-workers at King's College London. This single vague sentence was the only credit given, completely obscuring the fact that Franklin's data was not just stimulating, it was the foundational evidence upon which their entire model was built. Tragically, Rosalind Franklin would never have the chance to fight for her legacy. She left the difficult environment of King's College for the more collegial Birkbeck College, where she did brilliant work on the structure of viruses. But in 1956, she was diagnosed with ovarian cancer, likely linked to her extensive work with X-ray radiation. She died in 1958 at the age of just 37. 4 years later, in 1962, the Nobel Prize in Physiology or Medicine for the discovery of the structure of DNA was awarded to James Watson, Francis Crick, and Maurice Wilkins. Franklin's name was barely mentioned. The Nobel Committee's rules prohibit awarding a prize posthumously, so she was ineligible. But the debate rages. Had she been alive, would she have been included? The prize can only be shared by a maximum of 3 people. Given the historical and systemic gender bias in the sciences and the way her contribution had already been minimized, many historians believe it's unlikely she would have received it even if she had lived. For years, her story remained a footnote. It was only with the publication of Watson's own book, The Double Helix, in 1968, which contained a condescending and often inaccurate portrayal of Franklin, that a counter-narrative began to emerge, as friends and colleagues like Anne Sayre and Aaron Klug rushed to defend her and set the record straight. Today, Rosalind Franklin's legacy has been rightfully re-examined. She is no longer a forgotten figure, but is celebrated as the unsung heroine of DNA, a symbol of the struggle for recognition faced by women in science. Her story is a powerful and cautionary tale about the nature of discovery, the ethics of collaboration, and the importance of giving credit where it is profoundly and undeniably due. She was a brilliant scientist, a perfectionist in a field that demanded it, whose work provided the crucial evidence to unlock life's greatest secret. Rosalind Franklin's story is a powerful reminder that history isn't always written by those who do the work, but by those who tell the story. By sharing her truth, we not only honor her memory, but also inspire a more just and equitable future for all scientists. If you found this story as compelling as we do, please give this video a thumbs up, share it with someone who needs to hear it, and make sure you subscribe to our channel and ring that notification bell for more untold stories of scientific geniuses. Thanks for watching, and we'll see you in the next one.